Okay, second video. I'm going to quickly go through transformation. I apologize. I made this review for you a little lengthy. Wasn't trying to do that. But like I said, this is different than anatomy and physiology labs where I can say, know all these hormones, know their function, know what they do. These are experiments, and my time with you is a little bit limited. So transformation. So just to go back, this guide you have posted under tab number 7. So what you did, this was the experiment. You had 2 tubes. One was green, one was orange. You labeled one a positive and then one a negative. The first step, you took the yellow tube that had the transformation solution, and I told you to half it. It says here to do 250 microliters. If you remotely have any caffeine in the morning, and I'm highly caffeinated most of the time, that's almost impossible. That's why I said just divide it up in half. Okay, you put the transformation solution in. You then placed them on ice, and then while they were sitting there, you acquired the starter plates that had the E. coli on there, and you put the E. coli into both tubes. Right? Then you could put them back on ice. That probably wasn't quite as important, but as long as you got them sort of cold in the beginning. Step number 5 was you took the plasmid DNA in that little bitty vial, and you took the yellow loops and you put the plasmid in the plus only test tube. So only the cells in the plus got exposed to the plasmids. Now these Pigo Plasmid, this experiment, I don't know where it says Pigo everywhere, but that plasmids are circular double-stranded external DNA, and these are man-made. These are made, they're made in a lab, okay? And they have 2 genes. One's for antibiotic resistance to ampicillin, and the other gene is for a green fluorescent protein. So only the cells that get exposed to the plasmid are going to try to express those genes. So you added the plasmid. You then, I believe this is the point where it said incubate the tubes on the ice for 10 minutes. And then you had these 4 plates. One had LB, that stands for luria broth. I'll point that out. 2 of these were LB/AMP. That's luria broth/AMP is ampicillin. Luria broth just means that this is general media. It'll grow anything. It's just that's the kind of nutrients that were put into the agar. This one here, you had one of these plates that had 3 abbreviations: LB, AMP, ampicillin, and ARA, which stands for arabinose. You labeled these 2 positive, these 2 negative. So while these tubes were sitting on ice for 10 minutes, you did all this. At the 10-minute mark, you took the little floaty styrofoam things, that's very technical, you put them in a water bath, like I said, it's for 50 seconds, and then after 50 seconds, you put them back on ice, you see there, for 2 minutes. This is called heat shock. So let me just back up. The transformation solution that I had you divide up into halves like that, that makes a cell competent. And I'll point this out again. The term competent means that it's in a state to accept donor DNA. You made the cell more permeable. That's why you use that solution. This little heat shock step, wherever it is, there, and I'm going to go quickly to our review and I'll go back. This is a diagram that's straight out of the Biorad manual for setting this whole thing up. So this is supposed to be an E. coli cell. Bacteria cells have 1 single circular chromosome. Those little ring guys are supposed to represent the Pigo plasmids. They're floating in the tube with the cells that you added. When you heat shock, that going from cold to hot back to cold again, it forces, not all of them, but these plasmids move across, and where it says a pore right there, that means this is a competent cell. It's been soaking in that transformation solution. And so it's more competent to do this. And it's that change from cold to hot back to cold forces these little plasmids inside the cell. And the goal is, and what it's kind of showing here, the cell will transcribe it, then translate it, that's all in lecture, and produce these two proteins under the right conditions. This is a green fluorescent protein. This is an enzyme called beta-lactamase, which is antibiotic resistance. So that heat shock was kind of an important step. So to finish out what we're doing, so you did this, you went back, you put it on ice for 10 minutes, you added this luria broth to the tubes. Now that's for growth. That, like I said, that's media. That's basically food for the cells. You let it sit for 10 minutes at room temperature. You then took pipettes and you dropped, on the plates. It looks like this one was the positive vial. So you put basically bacteria on these two plates and you had them labeled positive like you did up here. This is the negative vial. You put bacteria on these two plates. You spread it around, you taped it up, and right now as we speak, they're baking at Howard College. So the goal is you want to make what are called some transformed cells. All right, so know those steps. Just kind of read through those steps of that experiment. So going back up here, so transformation is known as genetic transformation. It's when basically when you're genetically transforming a cell, you're making it produce a gene, express a gene that it normally doesn't do. It's been transformed. So the background here, a plasmid is defined as a double-stranded piece of external DNA, and you know what? I should have circular double-stranded piece of external DNA. They occur naturally in the environment. The one we used for this particular exercise was actually made in a lab. It's not natural. What they do normally is a cell will bring them across like this, and they usually carry what are called luxury genes. They help a cell out. They'll allow a cell to make a capsule, which is harder to kill, makes it more resistant to an immune system, or produce antibiotic, like down here, produce what's called these antibiotic resistance. They don't actually produce these green fluorescent proteins. Like I said, this plasmid has actually been made in a lab, so that's a special thing. All right, so this is just an example. So here's the nucleus of a cell. This is where transcription takes place. Translation is the process where you link together amino acids by reading the genetic code. And I picked this picture because it's showing these red dots. That's the products of the gene expression. In other words, the cell is reading some DNA, it's transcribing it, it translates it, and now it's made these proteins. Now I know that says toxic proteins, but that could be beta-lactamase, that could be the green fluorescent protein, if you follow what I'm saying. The plasmid in this experiment is called the Pigo Plasmid. It has two genes. You need to know this. It's got a gene that codes for beta-lactamase. That's the enzyme, ASE. Beta-lactamase breaks down ampicillin. So any cell that produces beta-lactamase is resistant to ampicillin. It's antibiotic resistant. So that's the antibiotic we use in this experiment. So it codes for that protein, okay? And then the other gene, it codes for what's called a green fluorescent protein. And it's basically the bioluminescent jellyfish that you see in the ocean. You can see them at night. Sometimes when moonlight hits them, they'll glow like this, and they glow that green color. That's the other protein that we're hoping is going to be produced. Now, here's the catch. This is very important. This particular protein, this gene for this protein, is only activated if the sugar arabinose is present in the agar/media. It's designed that way. Okay, so the designers of the plasmid, at the part where the cell's going to read this gene, they put a code in there that arabinose has to be present to activate the gene. So if there's no arabinose in the agar, then this particular gene slash protein, the gene will not be activated and a cell will not produce the green fluorescent protein, GFP gene. It's called the GFP gene. Maybe I'll put that in here before you guys get a hold of it. So, purpose of the experiment is we want to try to transform some E. coli cells. E. coli normally is killed by ampicillin. So right here was a little bit of a paragraph that was out of their manual. It says, what are we going to do during the experiment? I just went through that with you. Use transformation solution, calcium chloride. That makes the cells more competent. Carry out a procedure referred to as heat shock. I showed you that, where you go from cold to hot to cold again. So transform cells to grow in the presence of ampicillin. And then this is provide nutrients and short incubation period to expressing their newly acquired genes. Now you started that when you added the luria broth, then when we put it on the plates, and then now they're sitting in the incubator. So, and they have a nice little equation like just exactly like this picture I gave you up here. All right, so that's transcription, translation, and there's the protein. So they're saying here's DNA to RNA, that's transcription, to a protein, that's translation. And then you have a trait, which a trait would be antibiotic resistance, glowing green in the dark, that kind of thing. So know these terms. Transformation is a process which competent bacteria cells, ones that are permeable, pick up DNA from the environment and make use of the genes that says it carries. Okay, in this case, and I'll put in parentheses here, plasmid from the environment. The term competent means a cell that is competent is in a state to accept donor or outside DNA. It's more permeable. That transformation solution did that. It creates larger openings in the cell wall and the cell membrane. So the plasmid's going to get inside. Yeah, when you added the transformation solution in the beginning, the yellow tube, that solution is designed to make the cell more competent. A plasmid, there we got this one right, a circular piece of double-stranded DNA. Yeah, and once they're brought inside of a cell, cells transcribe and translate the genes on a plasmid, piece of DNA. Heat shock, I told you that. It's going from a cold environment to a hot short term to back to cold. That helps force, like it says here, the Pigo Plasmids inside the wall membrane, the competent E. coli cells. That's a critical step. The setup, just read through that guide I went through at the beginning of this video, please. And okay, quickly, I'll just go through this. This is the last part of this. Okay, when you get your results, this is what you're looking for. Now, I had this pulled up. You have this picture here. Here's the LB plate, which is a control plate. Here's the LB AMP with the negative, and both these got the bacteria cells from the negative vial. These never saw the plasmids. Remember, that was the negative vial. You added the plasmids to the positive vial. Here's an LB AMP with the positive. See, it was marked positive. Those are E. coli cells that were exposed to the plasmids. And then here's the one with the three abbreviations: LB AMP ARA. So those were marked plus, so you put the cells from the positive tube on these two. So you have that picture. All right, the LB negative plate. What is its purpose in life? We all want that. It's called a control plate. It is to test for growth, to make sure you have, like it says here, a healthy growing bacterium for the experiment. If you do an experiment and your plates all look like this one in the corner, no growth whatsoever, including this one, you don't know what happened. I'm just saying, you're like, oh, well, good grief, is it my experiment? What happened? But here, if you have growth on this plate here and then the others are blank, well, it's not the bacteria's fault. So that's why it's called a control, is to make sure you have a healthy organism. Something else went wrong. if your results didn't go right. I just put here the negative means that you put the E. coli cells that did not. I need to, yeah, just sort of underline that. There we go. I don't know. Maybe bald. Excuse me. Just thinking of my haircut. Maybe bold is too much. Underlining is good. These were the cells in the tube you labeled negative. So this plate is general agar, no antibiotics, no sugar. It will support anything. If you found something on the floor and swabbed it on here, it would grow, just like we did the first day. This plate should have growth. Okay, if you have a good experiment, a good organism, this plate, if you're asked, is this a normal result for this plate? Yes. The LB negative plate. Okay, you can see it's completely clear, you know, and that's where I got this. See that in the corner? No growth whatsoever. So this plate has the antibiotic ampicillin. That's why that abbreviation AMP is there. And that person writes much neater than I do. The LB just stands for the luria broth you added during the experiment, journal media. So it was mixed in, yes, when you make these plates, i.e. me, when you make this agar, there's a step where you pour all the plates to make these, the general ones, and then you add this ampicillin and you pour all these plates. So these plates have ampicillin mixed into the agar. If you swab the floor and swabbed these plates, very few things, if anything, is going to grow on it, because ampicillin is a pretty strong antibiotic and it's going to kill just about everything. The negative on this plate means that, yes, these were also the cells from the negative vial. You labeled it negative. So these were cells that never got exposed to the plasmid, like it says there. So these cells are not expressing any antibiotic resistance genes because, like I said, ampicillin, a strong antibiotic, kills E. coli. It's a gimme. And during this experiment, if you didn't expose any cells to a plasmid, they never got a chance to acquire it. They never got the chance to express the gene for antibiotic resistance. So this is a good result. This plate, like it says here, should have no growth. E. coli is killed by ampicillin, and it should be clear, should have nothing on it. And the reason is that's a normal reaction. E. coli is killed by ampicillin, and like I said, the fact that this is a negative plate, these were cells that never got the opportunity with the plasmid, never got the opportunity to bring them across, express the genes, and have a chance of surviving. This plate here, it's got the plus, it's got the two abbreviations, LB, AMP. It says this plate also has the antibiotic ampicillin in the agar. The plus means that you got the cells on here were from the positive vial during your experiment. You added cells, but then you also added the plasmids. So it says here, this plate got the E. coli cells that were exposed to the plasmids, and you added transformation solution in the beginning. You added bacteria. You incubated them. Yes. So anyway, I probably should put there, you added transformation. You added bacteria, and then you added plasmids, and then did the whole heat shock and all that. So on that plate, when you heat shocked, you had this going on. You had the cell, it's competent, and you had these little plasmid DNAs on some of the cells, not all of them. This is not 100%. In fact, it's a very low percentage, but some of the cells acquire this plasmid. So what you have on this particular example, all those little dots there, I can make that a little bigger maybe. Yeah, those are colonies. They're not cells. Okay, cells are very small. Those are tiny little colonies, and those are the cells that acquired the plasmid, and they're expressing the gene for antibiotic resistance, the beta-lactamase gene. So they survived, and now they've made colonies. And yes, millions of other cells got killed. The ones you dropped on there, this whole transformation process, this experiment, it's like anything in life. It's not 100%. Like it's a lower end percentage. Probably 20% of the cells got the plasmids. Okay, it's not going to be 100%. And this would be a good result. You should have, like it says here, somewhere on the bottom, this plate should have some isolated colonies growing if transformation took place. Okay? And in this case, if you were asked, did transformation take place here? And you see the plus, you see the LB, AMP, you would say yes, because those are cells that acquired the plasmid. They're transformed cells. Like it says there, any cells growing on this plate are transformed cells. And see, now you're able to select the ones that got transformed. That's the brilliance of putting the antibiotic resistance gene on the plasmid. If you looked at that plate and you're like, okay, well, which one survived, which didn't? The ones that survived are the ones growing. So now your experiment worked. That's how you know it worked. These are transformed cells. You're making them do something they normally couldn't do. They can't survive ampicillin. And it says that there. They're expressing the antibiotic resistance genes. Now, we'll get to do this in lab, but this little pin here is from Biorad. You push the button on the back, it emits a black light. Just like, anything fluorescent would glow, like the Halloween lights, that kind of thing. Well, what you could do, if you take this plate with the LB AMP, if you shine that light on this plate, it's not going to glow. It's going to look just like this, exactly like that. And I have another picture to show you to kind of hammer that point home. And if the reason why they're not glowing, it says it right here. When you shine a black light on this plate, these colonies do not glow green. Why is that? Because this particular plate does not have arabinose. You have to have arabinose in the media, the agar, for that gene to be activated to produce the green fluorescent protein. All right, so the reason these plates only have ampicillin, they do not contain the sugar arabinose. So in summary, this plate has cells only expressing one of the two genes on the Pigo Plasmid, the gene for antibiotic resistance. Remember, there was two of them. And it just talks about that right here too. So basically, and like I said, it's not 100%. Transformation is not going to affect every single cell. On this particular plate, you had millions of bacteria cells get killed by the ampicillin because they didn't acquire the plasmid. It's just not 100%. All right, last plate. This is the one with the 3 abbreviations. It's got the LB, which is luria broth. I actually had that up here in case you were asked that. Right there, LB equals luria broth. It has ampicillin and it has the ARA, which stands for arabinose. That's a sugar. And both of these, what you do is you pour the first plates with just the luria broth. You add the ampicillin. You pour about 16 of those. And then at the end, you add arabinose, and you pour like 8 to 16 of those kind of plates. So they have all 3 of them. The positive means that they're getting the cells from the positive vial. That's what that's saying here. You got E. coli cells that were exposed to the plasmid. Any cells growing on these plate are also transformed cells. And I just went over it again, redundant. On the dictionary it says, under redundant, it says C redundant. But nothing's 100%, so not every cell is transformed on this plate. Cells on this plate are expressing 2 genes. The gene for antibiotic resistance that codes for beta-lactamase, and it's also the gene for the green fluorescent protein. Now, if you're not shining a black light, they look like this. So that diagram, this one, see, look, they all look the same. If you turn the lights out and you shine the little Biorad UV light, the pen light on these, this one's not going to glow. This one will not glow. This one right here, I'm sorry, this one will not glow, but this one will. Why? Because it's got the sugar arabinose. Arabinose activates the gene. It's the only one that's going to glow in the dark. I actually, and I'm sorry I don't have this pulled up. I had it, where is it? There we go. I actually think I gave you access to this picture too, but so if you turn the light out, and this actually they're actually illuminating the plates as well, but here's the that says negative DNA. So these were the negative plates. See, this has no growth because it's got the amphisilin. This is the control plate. Okay, so of course those two aren't going to glow in the dark because they didn't get plasmids at all. This one in the left corner is the positive, and it's got the LB, A, and P at the bottom. But like I was saying, these plates only have one, the cells only have one gene being expressed because there's no sugar here. And you can see these glowing green. That's because there's sugar in the media. These are ones that survived amphisilin, so they're expressing that antibiotic resistance gene, but they're also expressing the green fluorescent protein gene. And I have a little bit picture. It really looks, so this is what it would look like without the black light. In real life, it looks just like that. And you turn the light out. You don't even have to turn the light out. You can have the lights kind of dim. But see the green colonies? You just flash that on there. So it doesn't have to be a bio-red light. Any black light would do this. And the reason this is very important is because why is that glowing green? Because it's expressing the gene that produces the GFP, the green fluorescent protein. And why is it doing that? Because there's sugar in the media, and that's activating that gene. So that's the reason that one glows green, and the one that was up here, okay, that had the positive and has the cells growing, but you can see by the abbreviation, this does not have sugar in the media. All right, media meaning agar. Now, I pulled up another. I gave you access to this picture, so this is a nice way to end this. And I apologize, this video is longer than I want it to be. So here's the little setup. These are drawings. So here's the plus plus, the minus minus. Here is the control plate. See how it's got, and that what you call this is lawn growth. When you have no colonies, that means it's super dense. It's like this is not lawn growth. You have individual colonies. That's why you do a streak for isolation, so you can get individual colonies and you don't have this big lawn growth thing. So this was the control plate. You're testing for growth. No growth, negative P glow. That's the way the experiment's supposed to work. These were cells that never got the plasmid, so they were killed by amphisilin. This one here, the LB, AMP, ARA, you see that it says fluorescent green colonies under UV light. That one has two genes being expressed by the cells. These are colonies, but the cells inside the colonies, they survived the amphisilin, because it's right there, because they got the plasmid, and they're also expressing the green fluorescent protein because of the sugar in the media. This one over here, this is the plus. You can see it has some isolated colonies. Those are cells inside. They are also transformed, but there's only one gene being expressed here because, and that would be the beta lactamase, the antibiotic resistance, because there's no sugar in the media, and the green fluorescent protein was not turned on, the gene for it. Okay, I hope these help. I didn't mean to make such a long review. But anyway, I wanted to make sure you had everything. So just read through this review a couple of times. You know everything on that review. You'll make 100. All right? Thanks. I appreciate you watching my video. Thanks.